Hello children, today I am going to continue with chapter Sexual Reproduction in Flowering Plants. The first event which occurs in the sexual life cycle of an angiosperm is development that leads to the formation of male gametes. And this topic further includes development of anther, that is the microsporangium, formation of microspore, that is pollen grain, and last, development of male gametophyte. Now, how does the development of microsporangium? Microsporangium is the pollen sac. Sporangia is sac and microspore means pollen sac. How does it occur inside the anther? Now remember, anther is a bilobed structure. Bilobed means it has two lobes and each lobe has two pollen sacs. So therefore, anther has total 4 pollen sacs. Now how does this development takes place? Now this development of pollen sac, that is microsporangia, begins with the differentiation of archisporial cells which are present just below the epidermis of anther. Below the epidermis we call it hypodermal. Hypo means below. Now, once this archisporial cells they get differentiated, then they start dividing periclinally. Periclinally means which is parallel to the tissues. So therefore, as a result of parallel division or periclinal division, the number of layers will increase, and the outer layer is known as primary parietal cell layer and inner is known as sporogenous layer. Now this primary parietal layer, which is just below the epidermis, will further undergo periclinal divisions, that is again will increase the number of layers, and there will be 3 to 5 concentric layers will be formed inside the epidermis, and these layers actually form the wall of the sporangium. Now this wall layer from the periphery is outside to center, consists of, now which is the outer layer, that is single layer epidermis. After single layer epidermis comes a single layer endothecium layer, and then comes 1 to 3 layers of middle layers, and the innermost layer of middle layer, that is known as tapetum, and very important function of tapetum is to give nourishment, that is food, to the developing pollen grains. Now the layer that I told you, that which is just below the epidermis, that is known as endothecium, its main function is that it helps in the breaking of anther, splitting of anther to release pollen grains when pollination it starts. Now here in the diagram we can see the structure of an anther. Anther, as I told you, is bilobed and each lobe has 2 pollen sacs. So therefore total sacs are 4, and here in this we can see the transfer section. Transfer section means the horizontal section where it is showing 4 pollen sacs or microsporangia inside the anther. Now let's see again what is the wall of this microsporangia. Now wall, first layer, outer layer is epidermis, then after that comes endothecium, second layer, then after endothecium comes 2 to 3 layers, and that we call middle layers, and the innermost layer of middle layer is known as tapetum, and inside we can see the sac which is containing microspore mother cells. Now next comes the formation of pollen grains or microspores. Now these are formed by the inner sporogenous layer, which is formed by the archisporial cell, and this inner sporogenous layer will divide many times to form microspore mother cells, which are the diploid cells. Now next, what happens? Each microspore mother cell will then undergo meiosis and it will form 4 haploid microspores or pollen grains. Okay, so as we all know that when a cell divides by meiosis, what happens? There is reduction in the chromosome number, and also what happens? 4 cells, haploid cells are formed. Now, once these microspores or pollen grains are formed, they remain associated. That means they remain in groups. So, group of 4 pollen grains is known as microspore tetrads, and different plants, they represent 5 types of tetrads on the basis of the arrangement of these 4 microspores inside these groups. So it can be tetrahedral tetrad, or it can be isobilateral tetrad. Another is decussate, inverted T-shaped tetrad, or linear tetrad. Now in each, you can see in each tetrad there is group of 4 pollen grains, and there is one plant that is Aristolochia elegans. In this plant, all these 5 different types of microspore tetrads are found. Now coming to the detailed structure of a single pollen grain or a microspore. Now as microspores or pollen grains, they are unicellular, means single cell. They have one nucleus, uninucleate, and that is haploid, as they are formed by meiosis, and they are spherical structures which further will develop male gametes. Now therefore we can say this microspore or pollen grain, it represents the first cell of male gamete. Now male gametes are not yet formed, they have to be formed. Okay, they are just containing a haploid nucleus. And what are the 2 layers of microspore or pollen grain? Outer is exine, which is thick, cuticularized, and very important, it is made up of a resistant fatty substance which is called sporopollenin, and no enzyme can degrade it, and it can withstand strong acids and alkalis. And that branch of biology which deals with the study of pollen and its exine structure is called palynology. The inner layer that is next to exine is intine, and intine is thin, it is smooth and cellulosic. Okay, and in the exine structure only, we can see there are gaps or points present in between the exine, and these gaps or points in the exine layer are known as germ pores. And germ pores, they serve a very important function, that is pollen tube emerges out of this germ pore. As we know, exine is thick, but the points or gaps where exine is absent, we call it germ pore, and this is the region from where the pollen tube will emerge out after pollination. And normally there can be 3 germ pores in dicots, and it can be 1 in the case of monocots. Development of male gametophyte, it starts when the pollen grains are still inside the anther lobes. So that means half of the development of male gametophyte, it gets completed before pollination. That means it comes to its 2 to 3 cell stage, and once it reaches its 2 to 3 cell stage, then after that anther breaks, releases the pollen grains, and then pollination will occur, and then further development of male gametophyte will take place after the pollination, and that will occur on the stigma. So that means this development occurs in 4 parts. That is, first is before pollination, it will come to 2 to 3 cell stage, then there will be breaking of anther to release pollen grains, then pollination will occur, and then lastly the further development of male gametophyte will occur after pollination. Now today I will explain you two parts, that is development of male gametophyte before pollination, and I will explain you till dehiscence of anther, that is once the pollen grains are released. Development of male gametophyte before pollination, it starts inside the anthers. Now during germination, what happens? The haploid nucleus of microspore or pollen grain, it gets displaced from the center to one side, and then this haploid nucleus will undergo mitotic division. Why mitotic division? Because it is already a haploid structure, and by undergoing mitosis it will form two unequal cells. Larger cell is vegetative cell and smaller cell is germinative cell. Now this vegetative cell, now this is two cell stage. What do we call this? Two cell stage, that is vegetative cell and germinative cell. Now their function is, germinative cell, it further will make two male gametes, and that too after pollination only. And vegetative cell, that also give rise to pollen tube, but this occurs only after pollination. And remember that in 60% of angiosperms, the pollen grains are shed, that is they fall at this two cell stage, and two cell stage means vegetative and germinative cell stage, and the further development of male gametophyte will take place only after pollination, and that will occur on the stigma. And in some cases, so these are the exceptions, that pollens are shed at 3 cell stage even in few of the plants, and thus in these plants the gamete formation take place before pollination. So this is only the exception in few cases. Next is breaking of anther to release pollen grains. Now as I told you just now that pollens, they get matured usually at 2 cell stage. Then what happens? They exert some pressure on the anther wall, and what happens? The wall of the anther will burst and will release all these germinating pollen grains into the air, and this we call dehiscence of anther, that is breaking of anther to release pollen grains. And in my next topic I will start with pollination, details of pollination, as well as development of male gametophyte after pollination. Okay children, thanks and stay safe.